Welcome to Mr. Chalk's Revision Tips. In this video, we will look at respiration, mitochondria, and So respiration is the chemical reactions that happens inside of cells that releases energy that they need to live. The reaction for it is glucose plus oxygen goes to form carbon dioxide and water. So respiration involves chemical reactions that break down nutrient molecules in living cells. Aerobic respiration needs oxygen, so it releases a relatively large amount of energy in cells by the breakdown of food substances. So if we look at the table below, it shows the percentage of some gases in the air that we breathe in and that we breathe out. So if we look at what happens in the lungs to change the levels of oxygen and carbon dioxide, some of the key things that you need to talk about is that oxygen passes from the air in the lungs to the body, carbon dioxide passes from the blood back into the lungs, and vice versa. So the substance with chemical formula C6H12O6 is glucose. If we're looking at naming the structures in the cytoplasm where cells carry out respiration, it's the mitochondria. If we're looking at what the waste products from respiration is, it's carbon dioxide and water. Aerobic respiration takes place in mitochondria within living organisms. So mitochondria, you can almost think of them as the powerhouses of the cells, because this is where respiration is taking place. So mitochondria are relatively small organelles. You might find several thousand of them inside of cells depending on what those cells need to do. So cells that are more active will need more mitochondria. So if we have a look at this experiment very quickly. So yeast can be used to look at respiration. So yeast can use other types of sugar instead of glucose. So some scientists investigated the effects of 3 different sugar types on the rate of respiration in yeast. Right, and they set up the experiment in that diagram. So if we were looking at 2 control variables that they'll need to keep the same, you're looking at something like the volume of yeast, the volume of sugar, the concentration of sugar, and the temperature would be important things to keep the same. If we were looking at making the experiment more accurate, you could repeat it more times. And if we're looking at why gas syringe is being used, it's to increase the precision of the results. Thanks for watching.